Hello, everyone. I'm here to present today. My name is Wang. I'm a third year PhD student in the bioinformatics department. I'm a member of the RAO lab, and I wanted to present on some of the tools I've used for my research into medical imaging, particularly a set of neuroimaging toolkits that have been helpful for wrangling some of the data, reformatting some of the data, and visualizing a lot of these radiology studies that I look at a lot. Um, so these toolkits that I'm going to be talking about today are part of the NIPI ecosystem. So the three toolkits I'm going to focus on today are nibabel, NILearn, and DIPI. These are tools that all come out of kind of a community of packages that are focused on neuroimaging, but I've also found them helpful for looking at medical imaging in general from different types of radiology. I've used them on prostate kind of studies because a lot of the underlying problems that come up in handling medical imaging data as volumes are also applicable to other um, applications of medical imaging. So I'd like to talk to you about these kind of packages today. And so roughly where some of the issues come in is when we're looking at medical imaging in general, one of the medical imaging is generally stored as DICOM files. And DICOM is a pretty good standard for certain things. It's built for radiologists, and it's how medical imaging is stored clinically. So anything radiology related from MR, CT, ultrasound, etc., is generally stored in the DICOM format on a PACS server, and can be. You'll see other things in there that radiologists put in, such as reports or derived images. And basically, all the DICOM file is an image and a header. And these headers are standardized in a way that you can store a lot of very useful information on how to read the image, how to interpret the image, and what the values are. But essentially, when you look at a MRI study, you'll get a stack of anywhere from 20 to 500 or many more, depending perhaps if it's a 4D volume of these DICOMs. And you put them all together, and that makes up your MRI study. And so all of this is great if you're a radiologist, but if you're a bioinformatician and you want to do something more involved with these studies, DICOM starts falling a little behind. It does. These headers contain everything from the where the each image sits in the volume. They contain oftentimes in the clinical setting, they'll contain PHI, and basically these headers are useful but not always filled out, and so that can sometimes cause an issue. And I'll give you an example of what some of these DICOMs might look like. This is a head MR from these are examples from MATLAB, and these are some. This is not the same study, but some of what the DICOM tags might look like. On the right, you'll see there's descriptions where it's taken. This one has anonymized version of the ID and that sort of thing. But there may be anywhere from 100 to there's tens of thousands of possible tags that are attached to headers, and they're also dependent on which scanner use these headers. And so that becomes sort of an issue when you're looking at these. This is a real example of some of the DICOM files I look at. This is just 25 for a particular modality, and they've been split out. But each one of these files contains an image and headers that are basically the same across all of the studies. Each time the patient's name doesn't change. The scanner that it's recorded on doesn't change. And a lot of these other files don't change, but the position of each of these files does change, and you need to put them together as a volume because MRI studies, the point of an MRI study is to look at it in a 3D sense. So you have to bring all this information together, anonymize it if you want to be sensitive to the PHI, and make sure you haven't lost any files along the way because the file names are not at all helpful. And that brings us to nibabel. I just wanted to Does anyone have any questions on the DICOM standard? I realize I should probably pause for a second and see if there's any questions. And if not, I'll go on to Nibabel, which is a package for handling a lot of these different imaging file formats, and contains a lot of the utility functions for these neuroimaging files. And so you might say, DICOM standard is great, and you could think of it as one of the image volume standards, but beyond DICOM, and once people start thinking, oh, maybe I need to actually handle images as a volume, you start running into issues. And there's a lot of different standards out there for how to handle files, as you can tell by this XKCD volume. So I'd recommend picking a particular one, and this is the one from Nibabel. Can handle a number of different ways that people store intermediate files, including DICOM. But I've used NIFT in the past. There's NRD. There's multiple different file formats that people use in order to store these MRI volumes in a consistent way for further processing. But you know, it doesn't really matter in the end as long as you're consistent across all of them and can handle different intermediate files. And so what is the Nibabel? Once Nibabel has taken in these files from either DICOM or NIFT or however you're storing these image files, it treats them as an entire image volume plus an affine matrix in order to handle rotations, shearing, and kind of changes to the image matrix. And this is important because DICOM itself is generally treats images as a stack, and so they're aligned, but they're basically the XYZ coordinate frame is only up and down, and it can't handle any rotations and this sort of thing, which is fine when you're looking at a single image volume. But once you start looking at images as a set of them, particularly an MRI, where everything's multimodality, where you're looking at a number of different aspects of the brain, and they're not always aligned properly. Having an affine matrix gives you a little more flexibility than simply a Z coordinate holding all the information about the relative spatial positioning of these different slices. So you can tell from this example on the right of you see that this is one slice out of one slice out of a brain a brain MR study, where you take your original pixels, which are in IJK coordinates, and transform them into real space millimeters meanings. So you go from this affine matrix tells you how to change those coordinates into something that can be properly aligned. And an affine matrix, which I'll be talking about later, contains both scaling, rotation, translation, and shearing. And one important thing to note is that these are all invertible matrices, which makes converting back and forth between these two formats, between these two coordinate systems, pretty straightforward, which is important. Next, I'll talk a little bit about some of the different, why this is important, some of the different neuroimaging MR sequences you might come across if you're working with MRI studies, particularly in the brain. You'll look at T1 images, T1 weighted images, T2 weighted images. They'll add contrast to an image to light up different. In a clinical setting, you may have a patient be injected with gadolinium or another contrast agent to study where the blood flows and that kind of effect, and see what changes are made when contrast is added in. Flare is a T2 type image where you're where you're doing inversion recovery in order to decrease the normal fluid and increase other aspects, but it looks sort of like a T2 image. You can kind of see I look at brain tumor images, which is not necessarily what all these neuroimaging packages are focused on, but is a reasonable application of these packages as well. And beyond these kind of standard MR sequences, you will also run into diffusion-weighted imaging, such as apparent diffusion. There's also an apparent diffusion coefficient, which is a secondary image where you've calculated a kind of a standardized scale of what this diffusion coefficient is. And there's many more, including perfusion, and I can go on and on and on about what type of sequences you're capable of taking in MR, which is something that distinguishes it from other modalities, imaging modalities such as CT or ultrasound, which are generally one acquisition and of the same type all at once. So that's part of why neuroimaging has a lot of these kind of packages for integrating different modalities. And so next I'll talk about NILearn, but in case anyone had any questions about NIBabel, I'd be happy to take them at this point as well. And if not, I'd also be happy to talk. I can't see the questions, unfortunately, but I was hoping if maybe they could be raised. But for the next, I'd like to talk about NILearn, which is a nice set of neuroimaging packages beyond the kind of utility functions that NIBabel provides, which also come with a nice, what I found very helpful in kind of these visualization tools and also other machine learning tools that are included in this is there's a lot of great documentation for this package. There is some good documentation for NIBabel as well, but it's less fully coded out in the way that NILearn is. And so some of the things you have access to with these NILearn packages, there's a lot of brain atlases. There's a lot of research into brain MR, particularly because of work in functional magnetic resonance imaging, fMRI. So a lot of normal brains have been atlased, and there's a lot of other data sets that come along with it. It provides a lot of good visualization tools for the brain, and there's a number of image manipulation tools as well. People have used these. I don't use it as much, but because there's a lot of work in functional magnetic resonance imaging space on finding which areas of the brain are connected to each other and their interactions with each other. This kind of human connectome project comes up. So there's a lot of tools in here. I won't specifically talk too much about that because that's not where my research focuses, but there are these kind of tools. And an important thing to know about fMRI is because it's an MRI which looks at a 3D space, but it also looks at this information and the change in blood flow over time, you're looking at four-dimensional data, which is always a little more tricky. Two-dimensional data is easier to handle than three-dimensional data, which is, again, easier to handle than four-dimensional data, which is always hard to visualize. So four-dimensional data is, NILearn provides a lot of these toolkits for handling this four-dimensional imaging data. And so some of the standard brain atlases you might look at are how we map out different regions of the brain and our study over time of how these different areas of gray and white matter are, what their functions are. You have motor cortex, that sort of thing. But I've shown two examples on the right. There's, in the example they've provided, there's probably like 10 to 20 different brain atlases you could pull down and map your brain images to. And so the idea is you take a structural MR, such as the T1 weighted imaging sequence, and map it onto this brain atlas. So you could use that to identify different regions of the brain in your study of a particular subject as well. And so a nice thing is there's a lot of visualization tools because it's difficult to take 3D volumes and present them as nice summary 2D images, particularly when you have both structure and then further on you have features you may want to record about these different areas of the brain. But you can see there's the standard kind of three cuts of you slice it down the midline after after mapping it to a particular atlas, and you can kind of see axial, coronal, and sagittal, these kind of three different directions of slicing the brain. But also there are ways of plotting these different atlases or different features or anatomy on top of that as heat maps, and different connections, which I find very appealing and also good for quality control, because it's important to, when you're looking at imaging tools, to make sure you know what you're looking at. It's easy to kind of assume that your pipeline worked correctly, but sometimes, if you forgot to align something correctly, or it's the layers of the mask don't line up correctly, it's just a nice sanity check to be able to take a look at a single image and say, this is what I'd expected to be investigating, and the values make sense to me. And also further down the line, if you need to present it to a clinician or in a paper, these are some nice imaging visualization tools. There's also a lot of image manipulation utilities I found for smoothing, resampling, masking. Oftentimes when you get in these different modalities, there will be at different resolutions, because it has different costs to sample at different under different protocols. So you'll need to map it if you have a segmentation done in one resolution, you'll need to resample it onto your different modality, either perfusion or whatever it may be, a diffusion mask, in order to make them all align properly, or smooth it, or handle some of these masking issues. And it does provide a lot of image manipulation tools as well. And again, these are all complemented by some nice Jupyter notebooks, which can give you some good examples on how to think about and handle this data, which I found very useful as well. And I mentioned that there's some Jupyter notebooks. There's a lot of them. This is just two of the sections that they provide, but you go to this page where they provide their examples. They have almost 100 examples of, or at least 50 examples of Jupyter notebooks, with lots of different tools for fMRI and MRI. And while it is focused on brain images in particular, some of the visualizations and tools, that's not, even if you're doing other projects, like I was looking at a prostate MR, still these kind of nice visualization tools can be utilized even if you're not doing the alignment or mapping it specifically to a brain. Structural MR or could it be CT or whatever you're using, still could be handled using these nice plotting tools. So that's, even if you're not using specifically neuroimaging, I think this is worth taking a look at. And so maybe I'd take another second to pause if anyone has any questions on NILearn. I'd like to also talk a lot about the next package I'd like to talk about is DIPy, which is Diffusion Imaging Python. So if anyone has any questions about NILearn, I'd be happy to hear. Maybe Aaron, if you heard anyone having any questions in the chat, you can bring them forward.
There's no questions yet.
Okay, it's all good. So I'll go on further to the diffusion imaging Python, and at the end, maybe if anyone has any questions or not, we can talk about that as well. But DIPy. So diffusion imaging is a different kind of imaging on top of MRI, where a normal MRI would, you're trying to see where the water is moving within, or the fluid is moving within a brain or a subject in general, a person in general, by doing this kind of diffusion gradient as well, where if it's, you'll take a pulse to set the hydrogen spins processing, and then provide a gradient and then flip the gradient, and then do an inverted gradient. So the idea is if you're staying stationary, these two gradients will cancel out. But if you've moved between these two time periods, if the water is moving, and particularly in a particular direction across these two time periods, you'll see a difference in the signal after these two paired gradients. And so you'll get out both diffusion-weighted imaging, which is the kind of signal you get out, and also an apparent diffusion coefficient, which is a single number you'd get out, a b-value that you'd get out to quantify what the diffusion levels are, as opposed to the rest of MR in general is not intensity based. The intensity is all relative. So apparent diffusion coefficient, the intensity does have a meaning, whereas most of the other imaging, including diffusion-weighted imaging, the signal intensity values doesn't have a meaning that is consistent across different acquisitions. So 100 in the diffusion-weighted imaging on one time doesn't mean the same thing as 100 on another image taken at a different time. But yeah, so what you get out from this movement of water is you get out a gradient field for the direction and the size of the movement of water and this diffusion across different time points, across the imaging sequence. And so what DI-Pi is kind of focused on is tracking these connections across these white matter bundles, and seeing, building out this kind of connection map across them, building these streamlines. I don't necessarily use it for this as much, but these are some interesting approaches I may look at in the future, but I use it more for some of the utilities that they provide. But it's an interesting way of looking at these gradient fields in this alternative way of looking at MRI imaging, and more information you can get out about the structure and functionality of these different brain structures. And so what I use out of it, the DI-Pi package, is mostly the image registration tools. Although again, there are a lot of diffusion imaging tools that are accompanying this. But in order to do this diffusion imaging kind of analysis, you'd first definitely need to do image registration. And so what is image registration? If you have two modalities of images that may be closely aligned but not quite aligned, you want to put them on top of each other so you can understand this multimodal imaging and really see, for a particular area of the body, what's happening in different non-invasive imaging metrics at the same time, right? So you might be looking at different contrast phases. You could be looking at totally different time points, or trying to account for trying to align it with a standard atlas, which is comparing it to a totally different person. Or you may be looking at different modalities from T1, T2. I mentioned all these other image modalities before, but if you can align them properly, and MR does some of it, but it's not perfect. And again, there are some caveats with it, but image registration helps bring all this together in a way that helps. You can analyze it properly. Again, 4D imaging, you also need to align it. And so this kind of static or this kind of affine registration, you can see on the bottom these are MATLAB tools. This is not exclusively through DI-Pi, but there are other tools that can do this as well. But you're trying to put these into alignment so you can either extend the image or provide some consistency across them. And so one of the ways that DI-Pi does this for affine registration, we talked a little bit about affine matrices before, is you're trying to find the affine matrix that brings two images into alignment, or two volumes into alignment that weren't in alignment before. using that 4x4 matrix we talked about earlier that allows you to do translation, rotation, scaling, and shearing. And so the way DIPy does this, the example they provided, and the way I'd recommend doing it is they go about it in different levels, right? So first, you want to handle the gross misregistrations because in any optimization problem, the closer you are to the correct solution, the better the fine tuning and your answers will be if you do this in steps. So it basically does, the way I'd recommend doing this is doing multiple sequential alignments, and they provide a nice tutorial for how to handle this. And the example they provided was you first align the center of mass. So if you have two brains, you want them to be roughly on top of each other first. And then you do a little bit of translation to make sure that you're trying to align them a little better than that. And then doing a rigid registration, which allows you to, while not shear it or change some of the structure of it, allows you to do rotation, scaling, and translation. And then finally allowing an affine registration at the end once you're pretty well aligned in order to properly map it. And again, each of these again has, you start at low resolution registration, fine tune it a little more, and then fine tune it a little more in this kind of pyramid structure where you start off with the gross structure and then fine tune it across these multiple steps. And the metric that you use to align these two images is often mutual information. That's helpful if they're not the same modality and not the same one. There's other approaches you could use, like squared error, root mean squared error, but mutual information is useful for cross-modality registrations because you're not dependent on them having the exact same value across the different modalities and volumes that you're looking at. And so here's this initial registration after center of mass registration. You can kind of see the shift. This is the initial alignment. And then here's the center of mass where you just put them on top of each other. And then you have a translation to do a little more alignment, because just because the center of mass is aligned doesn't mean it's properly aligned. And then finally, you can do rigid registration, where you kind of tilt it a little bit, and then a little bit of affine registration, where you get this nice, you can see with the green and the red are the two different, sorry, I didn't explain before, but the green and the red are the two different volumes, and you're trying to properly align them on top of each other. And you can see they're probably best aligned with this affine registration at the end, as they're more closely on top of each other. And so that's all well and good, but some problems don't necessarily fit under the affine registration framework. So in the brain, it's nice because there's not much movement, you're encased in the skull, and so things don't move around or slosh around too much. But in other problems, that's not necessarily the case, and you can't encapsulate all of the shifts with a 4x4 matrix. And that's where non-rigid registration comes in. And so non-rigid registration, this is an example from MATLAB, where this example is trying to align two hands on top of each other. It's the same rough structure, but there's been some movement akin to, say, heartbeat or breathing artifact you might see in another modality. But this allows for non-rigid registration, allows for individual shifts of individual pixels, and you're creating a kind of a mapping of the entire volume and seeing where different pixels are moving across it instead of a generalized 4x4 affine transformation where the entire volume is moving all at once. And so DIPy provides some nice toolkits for doing so. And you can see this kind of symmetric diffeomorphic registration, which is useful because, again, it's invertible, so you can have a forward and reverse kind of transform. It's less straightforward than an affine matrix, but the idea is similar, where you're kind of taking, if you have on the left, you have on the top row, you have the original matrix, and then you have on the right, you have a moving image. So you're going to shift it, and you can see where there's some shift, but it's not fully. You can do a further better alignment, and you can see that after you've warped it, they align better. But you could also, using the inverted matrix, inverse transform, warp the original image back using to make it match what the secondary image. Say you can make the red map look more like the green map with the inverse, instead of making the green map look more like the red map with the direct transform. And this uses a cross-correlation matrix and uses a similar idea, but I think this kind of, you can see the kind of shifting new coordinate system. This is a 2D image, but it's of course possible in 3D, and shows how this shifts happen. And to be honest, I think it's helpful. I would always make sure for these kind of image mapping, even if you did want to do a non-rigid registration, if you wanted to do that, I would also make sure you align them first in the kind of rigid sense. Make sure, because the further you have to move it, the more pressure you're putting on the system. So you can always do this in sequential steps and do an affine transformation first. And so other tools that are available in the beyond image registration, they're available in the DIPy package, are tissue classification, which is another problem that comes up often in the neuroimaging kind of setting. You want to know what, if you have, this is an example of, you have a T1 weighted image, and you want to know what the different parts of the brain are. From the classical kind of parts are gray matter, white matter, and cerebral spinal fluid, CSF. And you can kind of see this algorithm takes a T1 weighted image and kind of splits them into the different components. And this is not exclusive to DIPy. You always want to be careful, because depending on what image modalities your input set has, you can get different things with it. Some algorithms require more information, some algorithms and can get perhaps better results, unclear. But some algorithms require less information, and so depending on your problem, you want to tailor your solution to the proper level of. You may want to tailor your tissue classification algorithm to what you have. But there's also other things like algorithms they include, like skull stripping, to help clear out extraneous information about the skull and the other anatomy, and also do some anonymization. And these are all part of the DIPy package. And so the things I've learned about from these packages and the NI ecosystem in my work in neuroimaging in general is that someone probably has built the tool out there. It's not really worth reinventing the wheel. These are pretty solved problems. If you're looking for, you shouldn't be the one trying to, unless you have a specific reason to. If you only need something to strip the skull, or if you only need something to segment a tumor, or to do image registration, or whatever it may be, there's not a real reason for you to try and roll your own tool for a lot of these problems. It probably takes longer, and it's probably less well supported. So just take a look out there to see what kind of packages exist for image handling and pre-processing and visualization. There are some nicely documented packages out there as well. And I will say that there's a caveat that because everyone does things a different way and everyone has their own particular problem, while you may have my image data set may have standard MR sequences like T1, T2, flare and contrast, other people may be looking only at T1 and T2, and or they may expect, diffusion-weighted imaging, which I don't have, or um, a proton density weighted map that I don't have. You have to be careful to make sure that your inputs match what their algorithm expects. But it's worth at least looking around and seeing if you can find something that does a good enough job to get you the intermediate steps that you need. And even though I've mostly work for at least for my thesis, I mostly work in the brain MR space, I've also used these kind of tools in prostate MR or in liver CT, for example, and a lot of these radiological tools. And I think for those of us who are working in imaging in general, like their image registration is a good thing to think about for anyone who's working in multimodal imaging data. And there's a lot of good tools out there. And one of the things I've learned as well is that you got to look at the results. You can't just trust that everything works out perfect and pretty and nice. That's part of why there's these image registration tools to correct some of these issues that are relatively fixable, but you have to be able to see first that things don't line up. And if you just take a quick look at your data and make sure things line up correctly, you can save yourself a lot of hassle down the line with problems that are a lot harder to fix once you're looking only at numbers and matrices instead of the original images. For example, if you're trying to align brains to atlases in fMRI space, you're mostly looking at people who have normal brains, and brains are symmetric, and affine transformations work pretty well, and they work okay in the brain cancer space, but because there's a big tumor on one side of the brain is no longer symmetrical. So sometimes these algorithms of trying to align a normal brain to an abnormal brain don't work out perfectly. So taking a look at the images using image visualization tools from NILearn has proved helpful to me. So I just wanted to thank you all for coming and listening to my talk about my tools and tech presentation about neuroimaging toolkits and part of this NIPy ecosystem. There's a number of other packages if you're interested as well. But I'd like to thank you again and ask if anyone has any questions.
No questions. Thank you very much for your talk.
Okay, thank you very much for listening. I appreciate it, everyone.